My name is Mackenzie Taylor, and I'm an associate professor of biology at Creighton University. One really important way that we know information about prehistoric plants is from fossils. And fossils not only tell us what plants were alive where at certain times, they also give us a lot of important information about the biology of those plants. And different fossils can give us different types of information. So one really common type of plant fossil is a compression. And a compression fossil forms when plant material like a leaf or a stem or a flower gets compressed between layers of rock. And it gets pressed really, really fine, and all the water gets squished out, and it ends up being just a film of carbon. But this film tells us something about the size of the plant, the shape of the leaves, and sometimes you can even get information about the surface of those leaves, like where the pores or the stomata were, or maybe if there were trichomes or hairs on the leaves. Impressions are a fossil very similar to compressions, but in that case, all of the plant material, all of the carbon is gone, and you're just left with a negative imprint of the plant. But that also tells you really important information about the size and shape and surface of the plant organs. Some fossils form as a cast in a mold, and this is a three-dimensional reproduction of the plant. So the plant organ, like a root or a trunk or a seed, gets encased in rock, and that rock hardens around the organ, and then that is the mold. And then the plant material decays, and the mold fills up and hardens with sediment inside, and that's the cast. And the cast is then a 3D representation of what that plant organ looked like. And so you can get an idea of maybe where rootlets were attached or where leaves were attached, but there's no internal organization, it's just the surface in a 3D form. Sometimes plant fossils form when the plant organ is submerged in water, and that water is full of minerals like silica. And that silica gets inside the plant and deposits on the internal structures of the plant, like the cell walls. And then you can actually get information about what the cells were like inside the plant. So these are called permineralizations or petrifactions, like petrified wood. And these fossils are often thinly sliced and looked under a microscope. In addition to fossils, we can also learn a lot about prehistoric plants by building phylogenies, which are essentially family trees. Plants are often similar to their closest relatives, and so if we know what group of plants a fossil belonged to, like the pine family or the water lily family, we can hypothesize as to what a lot of their traits were like. And we can also look at the genes of living plants, and if we can reconstruct what the genes of their common ancestor might have been like, then we also can say something about what traits that plant might have had.